Let's dive in. This is an audio of the scientific article of the International Biology A-Level exam associated with questions 8 for the exam period October 2025. Fruit bats hanging on tree branches in daylight in Bangladesh on November 6, 2023. Some fruit bats eat up to twice their body weight in sugary mangoes, bananas, or figs every day to not only survive, but thrive. Unlike humans, these flying mammals can have an essentially permanent sweet tooth and do not develop some of the negative health consequences such as diabetes. A study published January 9 in the journal Nature Communications found that genetic adaptations have helped keep their sugary diets from becoming harmful. The study could have future implications for treating diabetes, which affects an estimated 38 million Americans, according to the Centers for Disease Control and Prevention, CDC. It is the eighth leading cause of death in the United States and the leading cause of kidney failure, lower limb amputations, and adult blindness. With diabetes, the human body can't produce or detect insulin, leading to problems controlling blood sugar, study co-author and University of California, San Francisco geneticist Nadav Ahatov said in a statement. But fruit bats have a genetic system that controls blood sugar without fail. We'd like to learn from that system to make better insulin or sugar sensing therapies for people. Fruit bats versus insect bats. Every day, fruit bats wake up after about 20 hours of sleep and feast on fruit before returning back to their caves, trees, or human-built structures to roost. To figure out how they can eat so much sugar and thrive, the team in this study focused on how the bat pancreas and kidneys evolved. The pancreas is an abdominal organ that controls blood sugar. Researchers compared the Jamaican fruit bat with an insect-eating bat called the big brown bat. They analyzed the gene expression, which genes were switched on or off, and regulatory DNA that controls gene expression. To do this, the team measured both the gene expression and regulatory DNA present in individual cells. These measurements show which types of cells primarily make up the bat's organs and also how these cells regulate the gene expression that manages their diet. They found that the compositions of the pancreas and kidneys in fruit bats evolved to accommodate their sugary diet. The pancreas had more cells to produce insulin, an essential hormone that tells the body to lower blood sugar. It also had more cells that produce another sugar-regulating hormone called glucagon. The fruit bat kidneys had more cells to trap scarce salts and electrolytes as they filter blood. Changes in DNA. Taking a closer look at the genetics behind this, the team saw that the regulatory DNA in those cells had evolved to switch the appropriate genes for fruit metabolism on or off. The insect-eating big brown bats had more cells that break down protein and conserve water, and the gene expression in these cells was calibrated to handle a diet of bugs. The organization of the DNA around the insulin and glucagon genes was very clearly different between the two bat species, study co-author and Menlo College biologist Wade Gordon said in a statement. The DNA around genes used to be considered junk, but our data shows that this regulatory DNA likely helps fruit bats react to sudden increases or decreases in blood sugar. While some of the fruit bats' biology resembled what is found in humans with diabetes, the bats are not known to have the same health effects. Even small changes, to single letters of DNA, make this diet viable for fruit bats, said Gordon. We need to understand high sugar metabolism like this to make progress helping the 1 in 3 Americans who are pre-diabetic. Studying bats for human health. Bats are one of the most diverse families of mammals, and everything from their immune systems to very particular diets are considered by some scientists to be examples of evolutionary triumph. This study is one of recent examples of how studying bats could have implications for human health, including in cancer research and virus prevention. For this study, Gordon and Ahatov traveled to Belize to participate in an annual batathon, where they took a census of wild bats and field samples. One of the Jamaican fruit bats that they captured at the batathon was used to study sugar metabolism. For me, bats are like superheroes, each one with an amazing superpower, whether it is echolocation, flying, blood-sucking without coagulation, or eating fruit and not getting diabetes, Ahatov said. This kind of work is just the beginning. Egyptian fruit bats on a fruit feeder at a zoo. After getting bit by a bat bug at a recent conference, Armin Shebin had a literal and figurative itch to study bats. The blood-sucking insect is one of many disease-causing parasites that latch themselves onto the flying mammals, yet bats rarely get sick in the same way humans do. Mammalian immune systems evolve fast as species are always challenged with new pathogens in their environment. You need to constantly keep pace with new bad guys that are trying to infect and hurt you, says Shebin, who is a postdoctoral fellow in population genomics at Cold Spring Harbor Laboratory, and has since recovered from the bite. And while he has studied the genetic adaptations of several mammals, they pale in comparison to the ones that have given bats the ability to fight off infections so effectively. In a new study published today in the journal Genome Biology and Evolution, Shebin and his team have identified the genes that have contributed to bats' rapidly evolving immune system and their unique ability to evade deadly viruses and even cancer. Understanding how bats survive diseases could inspire new immune treatments for humans and potentially help prevent another pandemic. The authors analyzed the DNA of 15 different bat species to get a clearer picture of how their genes evolved over time. They fully sequenced the genomes of two bat species, the Jamaican fruit bat and the Mesoamerican mustached bat, and gathered the other species from pre-existing datasets. They then compared the bat genomes to that of humans, mice, and other cancer-susceptible mammals, focusing their attention on the sequences that encode proteins responsible for causing or preventing diseases. To start, they lined up the homologous genes, or shared genes among different species inherited from a shared evolutionary ancestor. It's like comparing apples with apples, explains Shebin. With each homologous gene, they hypothesized two scenarios, if bats lost it or if it mutated. If the flying mammals completely lost the gene, it suggests that the omission is important in fighting disease. But if it remained with subtle changes in the DNA sequence that are only found in bats, it could show a change in gene function that somehow helps the group stay healthy. In the end, the most striking changes the team detected were in type 1 interferon, IFN, genes, which are important for controlling inflammatory responses to infections. Specifically, they observed a shift in the number of antiviral IFN alpha and IFN omega genes. For instance, 3 bat species seem to have lost all of their IFN alpha while increasing the number of IFN omega genes. According to Shebin, the most surprising finding was observing the loss of IFN alpha and addition of more IFN omega genes, which hadn't been reported at all before. The results suggest the new IFN omega and missing IFN alpha genes are important in bats for resisting viral infections while preventing overactive inflammatory responses, a feature that has made inflammation a double-edged sword in humans. But while the findings have put geneticists one step closer to understanding how bats evolved their unique ability to resist cancer and viruses, it doesn't paint a complete picture. The study focuses only on the genetics of innate immunity, the immediate immune response to infected cells, says Tony Schounts, a professor at the Center for Vector-Borne Infectious Diseases at Colorado State University, who was not involved in the study. It does not include information about bats' adaptive immunity, which consists of the antibody and T-cell responses that many mammals use to fight diseases. These are two very different but complementary components of immunity, Schounts explains. Nearly all of the focus on bat immunity to date has been on innate immunity, principally because the study of adaptive immunity requires live animals, which few groups have and is much more complicated. Even without a full set of information, understanding the changes in the bat's innate immune system could help scientists develop genetic treatments for humans that decrease susceptibility to certain illnesses. We can also learn which genes drive bats' 20 to 30-year lifespans, or how their bodies have adapted to process sugar-rich foods without developing the negative consequences seen in people with diabetes. And though bats have gained a notorious reputation for their purported role in spreading COVID, Shebin hopes that these new findings could point researchers in the right direction in understanding how the animals host such potent viruses and parasites without getting very sick. One day, he says, that information could be used to prevent our species from suffering major symptoms when infected. It's absolutely not misplaced to believe that studying bats could help us prevent another pandemic. This is the end of the article. If you are looking for notes, study guides, or potential questions in association with this article, you will be happy to know that by visiting www.tes.com you will be able to find any resource you wish for. We have our own online shop, hosted on the TES website. Our shop is your one-stop destination for educational excellence. So, what are you waiting for? Visit our online shop today and explore the resources available. Thank you so much for tuning in, and best of luck with your studies and exams.